see that our topics are arranged in such a way they flow into each other so that we are able to explain one, you have to learn one concept before you go to the next, so that you're able to understand as we proceed during the next academic year. And today we are looking at the cell injury as one of the initial requirements after looking at normal cell cycle, looking at what happens to cells when they're injured. Because our business as pathologists is to look at cell injury and the effects on the cell injury and how they actually look when they're injured by all sorts of various injurious agents. So today we shall look at the causes of cell injury. If you know a cause, then you can prevent it, or you might know how to mitigate whatever that is. The mechanisms of how these agents cause the injury. Then we shall look at the cellular changes at the light and electron microscopy. Most of our business is around the light microscope. It is possible to demonstrate some of the electron microscopy changes also under the electron microscope. And those are the very early changes before one is able to see them under the light microscope. It means that the magnification you see in the light microscope does not show the changes in the ultrastructural components of the cell. And therefore you need a more higher magnification as accorded by electron microscopy. Then we shall see what is necrosis from the pathology point of view, and a new concept called apoptosis, what that means, and the difference between it and necrosis, and what brings it about, and what is the intention for having such a process happening in the body. Okay, so we'll start by saying that cell injury can be due to a congenital issue, or it can be an acquired cause, or even part of aging. It is even the Bible and probably other religious book tell you that the lifespan of a man is 70, 80, and probably even up to 120, but with troubles thereafter. And that aging process shows similar features to what we see in cell injury. And therefore, when we are studying the cell injury and we are able to demonstrate what we see in cells caused by physical agents is also what we also see in aging. We see that in some of the body parts due to aging. Some of the acquired causes can be due to defective nutrition. You wonder where that nutrition come in. You remember in biochemistry, you learned about the food nutrients, the glucose, the building blocks of the carbohydrates, the amino acids, the nucleic, the nucleic acids are the building blocks for nuclear material. So the bases and what have you, and they are all derived from the nutrition that we take in. For some of these complex molecules to be made, like to make glycogen, you require enzymes, you require coenzymes, you require minerals, and therefore those are all part of the nutrition. Defects in nutrition or deficient nutrition, malnutrition will result in cell injury. You can even see that with the starving people, skin conditions and what have you, of vitamin deficiencies, you see skin conditions. Yeah, so defective nutrition, simply because all these nutrients are required for synthesis of the molecules that ensure integrity of the cell. Hypoxia, oxygen is required for the cells to remain alive, active. We need oxygen carried by the blood. So anything affecting blood, anything affecting the transportation of that oxygen Chemicals, either by accidental or day-to-day interaction or suicidal, and then drugs, which we administer as doctors. They're supposed to do a job, but we know that some create a problem. You take up aspirin, you get irritation of the stomach wall, you can bleed even to death if something is not done. Non-steroidal anti-inflammatory drugs can also do the same, yet you took them to mitigate some effects of the cell injury to begin with, but they have effects on other parts of the body. Physical, mechanical, during operations, as the surgeons are cutting, they crush tissue. So we end up with injury induced by some of the interventions that we have. Being crushed in a motor vehicle accident, you get injury of soft tissues, bone, which fractures. So even physical, mechanical factors, all those cause an injury. When somebody comes with a fracture, somebody comes with a stone in the bladder, compressing the bladder wall, all those are creating injury at the local site. Presence of a growth creates injury to the surrounding tissue. So there are many causes of injury. Microbial agents are part of it. And then we have immune reactions, infectious agents, as one of the main causes of people coming to see a doctor. They'll come to see you, and there will be something that will have initiated the condition that makes the patient come to see the doctor. And these are some of the causes that we are talking about. Cold, heat, people get damaged in a fire, tissues get cooked in hot air, the lungs gets damaged. That is now a physical type injury. And once the cell is injured, because we are living beings programmed in a particular way by the creator, we have the cells reacting, the ones that survive, the ones that don't die off from the injury, and they're in the neighborhood, in the vicinity of the injury, or it's a generalized type problem because it's an infectious disease which has become septicemic and is in the blood. Then even if you don't intervene, there'll be eventually the thing will come to a conclusion. And what happens is that the cells can either undergo resolution, where whatever was causing the disturbance is sorted out by the body so that everything is restored back to normal. We call that resolution. Or because if it's a continuous irritation, a smoker assaulting the respiratory tree with the continuous smoking, the smoke is an assault into the respiratory tree, then there'll be an adaptive change. Living in a polluted environment, you're inhaling things that are constantly irritating your respiratory tract, or if you're ingesting hazardous drinks like the changa, adulterated local brews, then you will also get irritation in the GIT, and you end up with an adaptive change. So adaptive change is to try to cope with the continuous irritation to the cells at a given site. The cells react by adjusting to cope with the new stress. And sometimes the assault is overwhelming and the cell can't cope, it can't heal and you get a resolution, it can't adapt and get a new appearance. What happens is that there's cell death. That becomes irreversible. It has been damaged to a point where the biological system does not allow it to recover. It just allows it to proceed now to a complete destruction, which we call cell death, and that is when you have irreversible cell injury. So it doesn't matter whatever the initiating factor is, you can either go through a resolution where things are restored back to normal, an adaptation when there's continuous irritation, or it is too much and the cell just dies off. Okay, so that's graphic. You get normal cells in the normal homeostasis. there's a disturbance by an injurious agent, which we said there are many, microbial, chemicals, whatever. It can be a reversible injury where the cells undergo resolution or an adaptive change, or it can be reversible where there's necrosis, the cell dies, or it can be programmed such that whatever physiological process brings that irritation leads to apoptosis, which we said we shall find out what the difference is between the two, because these are end results. But at this stage, we can go adaptive change or resolution, which is the desired effect. As doctors, we desire that there's reversible. We reverse whatever is disturbing the homeostasis so that the person can go on with their life. Okay, so what are some of the general principles we must consider? Is that when there's injury to the cells, the response by the cell depends on the type of the injury, the duration. Is it short-lived? If you dip your hand into hot water, as opposed to your hand being pushed and being kept there, the duration and the severity. Hot water might not be as bad as direct flame. So the severity of the injury. And that is the severity of the injury to the severity of injury to the tissue. Then we have consequences will depend also on how adaptive the cells are. Okay, there's the type of injury, the duration, and the severity. Direct fire is more severe on cells than water. But then, if you burn skin as opposed to burning bone or muscle, the consequences will depend on the type, state, and adaptability of the injured cells. All the cells don't have the same capability. They have different capabilities, and we shall see, depending on the capability to react to injury and regenerate, how cells adapt to injury or the effects of injury on the cells. And the systems that are thought to be affected during an injury, things you've already heard of, disturbance of production of ATP, which is required in the maintenance of integrity of the cell membrane. Cells are defined by a cytoplasmic membrane. A nucleus is also defined by a plasma membrane. The organelles have this membrane. If you damage that membrane, if for some reason that integrity is not maintained, because there's no production of ATP, which is required to maintain that membrane and maintain the differences between intracellular and extracellular milieu, then there will be a disruption and disturbance of the well-being of that cell. So if you interfere with the integrity of the membrane by interfering with ATP production, and if you interfere with the phosphorylation, oxidative phosphorylation, which happens in the mitochondria, then you interfere with the aerobic respiration, and the cell will not even be able to produce the ATP that will be required to maintain the membrane. So these things feed on to each other. And then you need protein synthesis. Protein synthesis synthesizes all sorts of proteins, structural proteins, enzymes, and all those that are needed for the well-being of the cell. And of course, the most critical, if there's disturbance in the genetic apparatus. The genetic apparatus contains the genetic code for coding for the proteins and everything else that is needed by the cell. And therefore, if you disrupt that engine, that's the end of that cell. In fact, you might survive some membrane damages, but once you damage the genetic apparatus, that's it. That's the core of the cell and what cannot be reversed. We also have some structural and biochemical elements of the cells that are closely related, that the structure affects the biochemical function. And we cannot tell at what precise point, either using the structure of biochemistry, we can't tell at what point that leads into widespread secondary effects. If we were able to, then we would design medications that can stop that. Subject of study, if you can define that point where cells become so disrupted and an individual gets so sick, then you would be able to come up with chemical molecules that can correct that or stop that, and you would be able to deal with many diseases. Because at the end of the day, each agent affects one of these factors and brings about the disease process that we see in the human being. Biochemical systems are thought to be disturbed earlier than the morphological structural changes, and that happens when there's quite critical biochemical disturbance for the structural disruption to also be displayed. So it is possible to demonstrate a biochemical change. If you do serum biochemistry, you will see a change before you see the structural change. It is possible to see that there's liver damage by doing liver function tests before we are even able to look and see that the cells have died off, or the cells have shrunk, or they have been disrupted. They are not looking normal. The chemistry can tell us earlier than we would see on histology. And then it has been shown that if you look for the intracellular organelles, you will be able to demonstrate changes earlier, even than what we see with the biochemical systems. And they are thought to be also to occur earlier before we are able to see the differences that we see on light microscopy. That the little organelles you see within the cytoplasm express disruption faster or earlier than what we see when we look at things under the light microscope. So if we are able to do electron microscopy, then we are able to see. How is that applied? In some kidney diseases and skin diseases, we are not able to demonstrate light microscope changes. But when we do electron microscopy, the person expresses a disease process. But when we look at it under the light microscope, we don't see. But when we look at it under the electron microscopy, we are able to see that there's ultrastructural component changes that could not be demonstrated on light microscopy. Light microscopy, the cells look okay, but when you go to the smaller components using light electron microscope, one is able to demonstrate changes. Okay, now we come to the concept of reversible and irreversible. If we can be able to catch things while they're still reversible, then there won't be much structural change or functional change in the individual. And these things have been studied and they're still being studied. And there are those that are defined as reversible changes that we can see on light microscopy, and the ones that we require electron microscopy. Under the light microscope, reversible changes show as fluid movement into the cell, causing the cell to swell. So that is called acute cellular swelling or hydropic change. So hydropic change in a cell is an indication of reversible cell injury. If you remove the injurious agent or treat the invading microbes or remove whatever mechanical pressure there is, then that cell recovers and continues to survive and function. And when we look at the electron microscopy changes, the reversible changes are shown as vacuolar degenerations. These are little vacuoles, fluid vacuoles actually, within the cytoplasm. And there's also endoplasmic reticulum dilatation. remember the plasma membrane that forms the ER, endoplasmic reticulum, and the ribosomes. This is the one where the ribosomes are attached. And in early reversible change, there's degranulation. The ribosomes disengage from the reticulum. And if they are polysomes, they disaggregate into single ribosomes that we can see on electron microscopy. If injurious process is stopped, these things can be reversed. If not, then one proceeds to other things. Examples of where we can see some of this is during ischemic change. Remember blood reduction of, we shall be looking at what ischemia is. Ischemia is the reduction of blood flow. If there's no effective flow, the cells will not get adequate oxygenation through the oxygen carried by the hemoglobin. They will not get enough nutrients, building blocks, which are within the blood, and therefore you won't get what is required for the production of ATP. And what happens is that there will be reduced ATP production, and the cell now is forced to switch to anaerobic glycolysis. It is okay for the cells that can do that, but we shall find out that there are some cells which can do that, or you already know, or you'll be learning about that. And then when there's a reduction in ATP, the sodium potassium pump fails, and fluid is not kept out, and therefore there's a net flow because the osmotic pressure in the cell is higher. And that's how we end up with the cell swelling, the one we talked about, hydropic change. And if that happens, also the other organelles with the plasma membrane, then there's also breakdown. And when there's breakdown, it continues drawing in more water. Of course, some of these injurious agents have a direct effect on the plasma membrane. Remember the MAC membrane attack complex? You're second year, so you might not know that too well. Immune responses result in these antigen-antibody reactions, the immune complexes attaching to the cell and creating a channel through which fluid can flow into and therefore disrupt. Yeah, or heat cooking the cell itself. So that's the direct disruption. Cutting through, that is direct cell disruption of the plasma membrane. What are some of the changes we see on some of these surfaces? There's a loss of microvilli. The cells that show microvilli, like in the GIT, there's a loss of those ones. Then you start getting protrusions, which are called surface blebs. It's like it forms protrusions that are called surface blebs. And we start getting disaggregation of myelin and forming what we call myelin figures. This is derived from the plasma membrane. So you see these myelin figures within the cytoplasm. You require the electron microscope to demonstrate, or special stains to show that. And that is because the lipid membrane, the plasma membrane, is degenerating and the components are aggregating into what we call myelin figures. What are some of the nuclear changes that one can see? Initially, there's mild clumping. These are the reversible ones. There's mild, usually it's a fine homogeneous staining with the hematoxylin and eosin, especially the hematoxylin part. And there's mild clumping so that it starts looking particulate. That's what we talk of clumping. The chromatin clumps, that is the DNA material clumps. And there's the separation of nucleolar. The nucleolus is as an organelle where you see a rounded body in the normal cell. But now you stop seeing it that way because the nucleic material in the nucleolus disaggregates into a fibrillar and granular components. You don't see it as a nice membrane-bound organelle. Instead, the components disaggregate and form fibrillar and granular depositions that you would have to demonstrate that was the organelle. It was actually the nucleolus. Okay, that is a reversible. What we see under the light microscope, and what if you were able to subject the cell to electron microscopy, one is able to see. How about now the stage where we say it is irreversible, the opposite of reversible, irreversible. It's been shown that you don't have a wall dividing the two or a clear-cut line, but it is a continuum. You move from reversible to irreversible, a continuum, and can't say it is at this point, this cell is at this point, this disease process, we are seeing these cells are not going to recover, all these are going to die off. We cannot tell that. It's not predictable. You cannot predict. We can only demonstrate when it has happened. But what do we see when it has happened then? We said in reversible, you see vacuolation. But now in irreversible, there are even more. Severe vacuolations, and we also see in the mitochondria flocculent densities, densities within the mitochondria that are usually not there, and that has been shown to be one of the most reliable early signs of irreversible injury. That if you look under the microscope and you look at the electron microscope and you look at mitochondria, you will see flocculent densities that indicate that the cell has been irreversibly injured and is not going to recover and is headed for necrosis. What do we see in the plasma membrane? That loss is more obvious. There's even loss of intracellular substance into the extracellular space. And of course, what that does is that you get the fluid coming in, and eventually the whole thing gets disrupted, dismantled, and the structure collapses completely. You don't see the membrane-bound structure. Instead, you see just ghosts of what was there before and not the actual cell. It's like disrupting a fence, and a house can be approached by anything. So this membrane gets disrupted and the extracellular material diffuses out, and you stop seeing a well-defined cell. Okay, in the cytoskeleton, we have some changes that include breakages of the microfilaments. These microfilaments give the shape of the cell. Some cells are rounded, some are oval, but generally some are columnar. So the shapes are given by these microfilaments and the intermediate filaments, and when they get disturbed, the shape of the cell is also disturbed. And that's why you end up with protrusions on the surface, forming what we call the surface blebs. You see rounded little bodies on top of the cell, and those ones we call blebs. Lysosomes also have a membrane binding them, and when it breaks, there's a release of all those enzymes. And within those enzymes, there's you have all sorts of enzymes that can digest every component of that cell. And given time, you will even stop seeing the cell components because they will have been digested down and reabsorbed as useful molecules to be reused in the synthesis of other cells or structures. Unfortunately, when these enzymes are activated, they will then digest the surrounding cells. There's digestion of the surrounding cells, and that creates havoc because it started as one cell and now it spreads, and that's why you end up with a wound or a necrotic site because of digestion by these dying cells. by the enzymes that have been released from the lysosomes. When we reach the irreversible stage, it doesn't matter what the initiating factor was, they all end up looking the same because it results in disruption of the structures and release of lysosomal enzymes, which eventually digest the cells, and that cell is lost completely. If it spreads over a wide area, then we see a necrotic bit within the organ. What are some of these biochemical mechanisms involved in the disruption of the plasma membrane? Depletion of ATP. ATP is required, it's an active process. Production is an active process. It is required to maintain a plasma membrane. It's a common effect in ischemic and toxic injury. Why? Oxidative phosphorylation can't happen, and therefore there's reduced ATP production. We also have production, there's a production of reactive radicals and reactive oxygen species which damage the surrounding structures, and that happens when there's generation of energy, and these are produced as byproducts. They activate the radicals, and then the radicals are important in the destruction of cells. It was a very important component in the damage that was being seen in COVID. COVID was damaging to the lungs, and part of it was because of this production of these radicals. Obviously, they are produced as a byproduct of the normal respiratory process. And when we have reduced oxygen, then we have quite a number of them being produced, and they now cause damage in the tissues where this is happening, like where there's ischemia or hypoxia, whatever is happening. Then you get damage by these reactive oxygen species. The other thing is that the body has protective systems that mop up these reactive oxygen species, but if they are produced in excess, the body systems can only cope up to a certain point, then they give up, you know. So these oxygen radical species are mopped up in the normal metabolism, but when there's injury, they are excessive and they overwhelm the system, the mop-up system, and therefore we talk of oxidative stress. And that also contributes to the further damaging of the cells. The story of calcium. Calcium as an ion is important in maintaining a lot of the body functions, and therefore we have when the homeostasis is disturbed, we get damage, especially to the membrane. It results in membrane damage and activation of enzymes. It's usually a core factor in enzymatic processes. So when there's general disturbance in calcium metabolism or homeostasis because of the injury, then you find that there's a disturbance of all these processes, the membrane, the enzymatic activation, and then also that calcium is needed by the ATP-dependent pump. And therefore, there's disruption of that pump, and part of the biochemical processes that contribute towards the effects that we see when there's cell injury. For some reason, if there's a defect in the membrane permeability, where there's a loss of early selective permeability, where water is kept out, then that means that there's water is drawn in, dilutes, interferes with the intracellular homeostasis, and also contributes towards the damage to the cells. Just to remind you that all membranes are involved by the injurious agents, especially the infectious agents, the complement, MAC complex, and also physical and chemical agents. Obviously, just the way we cook food is the way cells are damaged if you subject them to heat or if you subject them to extreme cold. There's direct damage to all the membranes. The cytoplasmic membrane, the nuclear membrane, they all get damaged. And that's why you end up with necrosis when there's frostbite. When somebody is subjected to extreme heat, the skin peels off. Even a sunburn is enough to cause skin peeling off. Okay, toxins target the enzymatic process in the mitochondria and causes irreversible damage, because we said one of the early changes of irreversible cell injury relates to mitochondrial oxidative phosphorylation stress. When we see the changes within the mitochondria as one of the earliest signs of irreversible change. So toxins, which affect the mitochondrial function, the cytochrome system, and here we're talking of hypoxia and toxins. And remember, drugs can also be toxins. They can be, some of the drugs can be toxic to the system. In fact, chemicals can be, and this actually damage, and therefore there's no oxidative phosphorylation happening. Then you go back to ATP, membrane integrity, and that leads to irreversible cell injury and cell death. Oxidative stress results in breakdown of phospholipids and an increase in cytosolic calcium and lipid breakdown products. And we said that irreversible mitochondrial damage is synonymous with irreversible cell injury. So when you get mitochondrial damage, that is synonymous with irreversible cell injury. Those we can't see on light microscopy. We have to do electron microscopy to be able to see mitochondrial damage. It becomes important when you have diseases where you can't see much gross changes, but and even light microscopic changes, yet the person has a disease process. In the kidney, there won't be much physical change, there won't be much histologic change, but they'll be having functional issues. They'll be having a failure, as exemplified by how they present. But we are not able to demonstrate that, and until we do electron microscopy, biochemistry shows there's a derangement. Grossly, we don't see anything. At microscopy, light microscopy, we don't see anything. Yet we know the patient has this clinical condition, and we can only demonstrate it because such changes are at the mitochondrial level or other organelle level. Okay, so that is in graphic form. ATP production and maintenance of the membranes, very important. Otherwise, when there's interference, you end up with necrosis. If you cannot produce ATP, as in when there's hypoxia and ischemia, then the end result will be a cell necrosis. When we have multiple injurious stimuli resulting in production of reactive oxygen species, then what happens is that there's direct damage to the lipids on the membranes, and that results in cell injury. It can also affect the proteins, structural and enzymatic, and or even affect the nucleic acids and lead to necrosis. Okay, you can get mutations, that is congenital, no, acquired. This can be acquired, or even cell stresses and infections, and those result in abnormal proteins that can cause apoptosis, which we haven't talked about yet. So we shall be talking about apoptosis later. Necrosis and apoptosis, we shall be talking about them later. But the end result, the cell has died and cannot go back to its normal functions. Most of our patients come because of infections or immunological disorders where there's derangement of the immune system. Many diseases have been shown to be because of that, and they end up with necrosis or apoptosis, depending on the disease process. The cells produce cytokines, which are damaging to the surrounding cells, and these are usually the lymphocytes and the leukocytes. They're usually the leukocytes, and they produce toxic molecules that damage the cells and lead to death or apoptosis. Okay, so that leads us to the next thing. And the next thing is necrosis. Then you ask yourself, you thought you knew what this word means, but today we shall tell you what it means. People say, oh, he died, he had a necrotic wound. What are you really talking about? A rotten wound, what are you talking about? So necrosis is a scientific word. It actually is a spectrum of morphologic changes that are seen. Here you have to take note, morphologic changes that are seen in the cell that lead to cell death, and it must happen during life. So there's necrotic tissue in a living person. We don't talk of necrotic tissue in a body, a dead body. We don't talk about that. That is decomposed. But when you're talking necrosis, it must occur during life, where there's morphologic changes resulting in the death of the tissue, but the surrounding tissue is living. So it is death of cells during life and not after death. Whatever happens after death is not necrosis. It's autolysis. We call it autolysis. The body autolyzes, everything stops, everything falls apart. The lysosome release the enzymes, and the enzymes digest everything. If you don't do anything to the body, to be digested, and then eventually nature will take care of it. There's the autolysis where the self-digestion. But here, there's injury where cannot maintain the cell homeostasis, and therefore you get tissue dying surrounded by living tissues. So a spectrum of morphologic changes following cell death in living tissue. That's the catchword, in living tissue. We don't talk of autolysis. Here, we are talking of necrosis in the living person. But the processes are similar. The same enzymes do that work, but here it gets limited because the surrounding tissue is living and it contains that process of autolysis. Whereas in the dead body, it continues and can digest the whole body. And this results from progressive degradation of enzymes on the lethally injured cells. They just don't remain there intact, but the body deals with it, because we said this is a biological system. They are plastic. They have to move to the next stage, next level, next whatever. They just don't remain where they are. They have to progress to the next thing. This can be a rapid process or a gradual process. When we say it is necrotizing, a necrotizing disease, diphtheria is a necrotizing throat infection, Clostridia is necrotizing lung, intestine. So bacteria can be very necrotizing, but we also have others where you get ischemic changes and there's necrosis, but it's all occurring in a living being, not the dead. I hope you understand. If we cut tissue and leave it on the bench, it will undergo autolysis, not necrosis. So necrosis we reserve for the living, where there's tissue dying because of a disease process. It can be rapid or gradual. And if it's because of reduced blood flow, this is another topic we shall do, for whatever reason, that the blood flow, remember blood brings nutrients, maintains, removes all that's being broken down. If that blood flow stops in a part, because if it's the whole body, the person dies. If it's in a part of the body, then we talk of infarction or ischemic necrosis, as we shall see that. How do we recognize that? And we're talking about necrosis because cell injury leads to death of part of the body that is injured. Somebody with a boil, where the pus is, that is dead cells. It is mixed with the pyogenic bacteria, Staph aureus, but it's not the only one. It has destroyed, necrotizing, caused the necrotizing inflammation of whatever site that is. And so we talk of the changes that we see, and the changes that we see in the cell. When we look at the cytoplasm, it appears eosinophilic, very pink compared to the surrounding tissue, and it has a glassy, homogeneous effect. A normal cell has vacuoles, so it's not homogeneous. It has differences in the way the cytoplasmic contents look. Some look paler, others look pink, but now this one is evenly pink and it's glassy in appearance, meaning that everything has been digested, and so everything is taking the staining in a similar way. The enzymes have done their duty, have digested the dead cell, and what remains there is just the breakdown products, which take up the hematoxin, the eosinophilic eosin. They take up the eosin, and so we talk of eosinophilia. Eosin. Eosin is the cytoplasm, the one that stains the cytoplasm. So we talk of eosinophilia, meaning that it takes up more of these stains. Philia is taking up more. So it takes up more of eosin in the H&E, and therefore it appears pink and glassy. Okay, we can also get a vacuolated appearance at the edges of the cytoplasm, the cell boundary. So we talk of a vacuolated, moth-eaten appearance, as if bits of it have been bitten off, but that is because of the fluid that is there. There's no cytoplasmic membrane, and so we talk of moth-eaten appearance, vacuolated moth-eaten appearance. That space is actually where the fluid was, and that's why we call it vacuolated moth-eaten. And with the time, the body does not like dead material just coexisting with living tissue, so it undergoes another pathologic process that you shall be learning about, called dystrophic calcification, where there's calcium deposited onto this material, and therefore it becomes calcified. So if somebody has TB of the node, and the node has necrotic material because of the bacteria, then it becomes calcified and might look like a stone. Yeah, so we talk of dystrophic calcification. Dystrophic to differentiate it from what happens when it occurs on normal tissue. So dystrophic, it is occurring on dead material. Okay, so that is a pap smear, and it shows a lot of neutrophils which have been attracted to the site, and within it we have the epithelial cells, which are showing some of the changes that we are talking about. You get a rounding off and cytoplasmic vacuolations in the cells that are about to be injured, and then we start seeing naked cells. We shall come back into that story. Okay, so when we have the necrosis, we end up with the amorphous material without any appearance. This is where now you have the cells which are being injured, and some will become dismantled. You can barely make out the cytoplasmic outlines of this particular cell. I don't know whether you are able to see my arrow. Let me make it a laser pointer. Okay, if you look at this cell, you can see the cytoplasmic boundaries here, but you can't see the boundaries here. So there's a release into the surrounding because we have bacteria, and the bacteria has caused an inflammation in these cells, and some of them are beginning to be disintegrated. They have started disintegrating, and the others that have already disintegrated, and we only see little fragments of the nuclear. And like I said, I'll come back so that we show you some of the things that we see when there's necrosis. Necrosis can also occur because of bacterial and fungal colonies. This is bacteria growing in normal tissue, provoking inflammation and necrosis within it because the cells break down and they are digested and become homogenized. Okay, if we look at it grossly, this is a lung, and within the lung, there was a necrotic tuberculous cavity, and the TB has destroyed all the normal lung, and all we are seeing is this necrotic material within that has a different appearance from the normal living. So this is necrotic as opposed to autolysis in a person who dies, because the rest is living and this one is clearly defined. Okay, how does it look on microscopy? We said it appears amorphous, and this is how it will appear. We have cells out here, but in here you have necrosis, and it's you can see nuclear debris. We are seeing some nuclear debris, and we are seeing the pink cytoplasmic contents, which have become homogenized and become, because this is more extensive, it's not a single cell, they join up and we see confluent necrosis. We call it confluent necrosis because the cells have broken down and all the cytoplasmic and nuclear contents have spilled out and are breaking down. We can still see the outlines of some of the nucleic material because it has taken a bit of hematoxylin, and the rest is cytoplasmic because it has taken eosin. And so we talk of eosinophilia and basophilia, or breakdown contents of the nucleic material. It can also be tumor causing necrosis, not necessarily an inflammation. It can also be tumor, so different causes of injury, bringing about necrosis of the surrounding tissue. This was a tumor in the mouth that was causing severe necrosis, and we can see the salivary gland tissue in the neighborhood, which is still viable, the tumor, and it has caused damage to the salivary gland tissue, which was here. Okay, I think we had already talked about that. Okay, now we were talking about necrosis and the morphologic appearance, and I showed you a few images. So what do we see on electron microscopy in necrosis? The same thing as we had seen in irreversible cell injury, dissociation of plasma and organelle membranes, dilatation of mitochondria with amorphous densities, and also eosinophilic debris, the amorphous eosinophilic debris and fluffy material within the mitochondria. And now those are the cytoplasmic changes. When we come to the nuclear changes, when it becomes necrotic, we have a little bit more that happens. And what is that? We get fading basophilia. The nucleic material starts breaking down, and therefore it doesn't take up the hematoxylin as strongly as it would, and because it appears blue, We call it basophilia, but now this is fading. It becomes lighter, not so intense. And that is because there's breakdown, which we call karyolysis. There's breakdown of the nucleus. The nucleus is the caryo part, and the breakdown is the lysis. So we have the breakdown of the nuclear, which is called karyolysis. And because of that, when we use hematoxylin and eosin, it takes the stain less intensely, and so we talk of fading basophilia. It is fading because you don't have all the nucleic material that should have been there, and therefore it's not taking the stain as it should. If it continues being broken down, lysed, what we end up with is picking. Okay, another is karyorrhexis. You can get complete dissolution of the nucleus, or you can have it breaking down into pieces, so that we call it karyorrhexis. Little fragments, like the ones I was showing you here, little fragments of the nucleic material around here. Here you don't have the blue fragments. Here we have karyolysis, karyorrhexis, and here in some areas we have complete karyolysis. You can't even see the nucleus or any hint that there was a nucleus. But here we see the blue, the fading basophilia, where we have part of the blue staining contents, which are due to the karyorrhectic nucleus, the breakdown product of the nucleic material. So here it breaks down into small fragments. Here it dissolves and completely goes off. So it starts by reducing in intensity and eventually disappears completely, where you can't even see that there was a nucleus. But it can also shrink and become very small, so that all the nucleic material shrink into a small nucleus that is called pycinosis. So we say that the nucleus is pycinotic, as opposed to the one that disappears or the one that breaks down into small pieces, karyorrhexis. And this is what I was trying to demonstrate in this other picture. In the squamous epithelium of the cervix, this is the cervix, the pap smear, there's shedding off of the superficial layer, and the basal ones look this big, but by the time they're coming to this stage, they're pycinotic, because it dies off as it's getting shed. On the skin, it even loses the nucleus completely. So we talk of squames, the cytoplasmic content with a membrane, but no nucleus that can be seen. Those are called squames. But when we have them very shrunken, like this particular nucleus here, then we talk of pycinosis. But this is how they start, and then they get injured and are shed to the surface, and they end up being pycinotic. When they become karyorrhectic, they are broken down like this, where you just see fragments. You see fragments of what was a cell here, and the residual nucleus. You just see the residual nucleus. You don't see what its cytoplasmic content were, but then you see another one which broke down into, this is a bare nucleus, but this other one broke down into karyorrhectic components because of the inflammation. Okay, that's when it breaks down and eventually disappears. And the necrotic material, like the one we were seeing here, can acquire several forms. Here, it's just a tuberculous cavity with necrotic material. This is usually what is coughed out and somebody is left with a cavity. But now, if it's in organs, we can have three forms of that. We can have a coagulative necrosis, where the outline remains, the outline of the cell, the cytoplasmic outline remain, the cytoplasmic, the shape of the infected area remains, and it can be preserved for some days until it is completely digested. When you look at it under the microscope, we see ghost outlines of cells. We don't see the nucleus, but we see ghost outlines. When we look at it as the gross specimen, we see the shape is maintained. If it is an infected part of the kidney, we shall see that wedge shape remaining within the kidney. But with the time, it gets digested by those lysosomal enzymes, as we have said, and they destroy them completely, and everything gets dissolved. But we have others which don't retain the shape. Instead, they go directly into digestion and liquefaction. So we talk of liquefactive or colliquative necrosis. And this is because like the brain has a lot of fluid content, it's a semi-solid organ. So when there's necrosis there, it undergoes liquefaction and forms a fluid cyst. We see fluid within it. If we have an infection of a bacteria, pyogenic bacteria, like Staph aureus, and you get a boil, then it also forms a pus, which is a liquid that contains bacteria and lysed tissue. So liquefactive also includes when we have pus formation. In the TB, tuberculosis infections, the material is thick and cheese-like, like Greek, what do we call it? The Greek cheese, and it appears caseous, and caseous means that it's cheese-like. And I have shown you with that image the way it looks quite thick, like you can rub it off. And that's what is called caseous necrosis. It's a type of coagulative necrosis unique to tuberculosis. We don't see other disease processes producing caseous necrosis, but it is also a coagulative necrosis unique to tuberculosis. The fat can be injured and produce fatty necrosis, and fatty necrosis is also a coagulative necrosis, but unique to fat. We can also get a scenario where there's necrosis, and then because the necrotic tissue is dead, we say that is irreversible injury where there's cell death. What happens is that bacteria colonize. The bacteria come in and colonize an area with coagulative necrosis, and when that happens, we call it gangrenous, and it can be wet gangrene or dry gangrene. And these bacteria that come are usually saprophytic. They are not the usual pyogenic disease causing. These are just bacteria that colonize rotting material, and therefore this is the necrosis is dead material, and therefore they come colonize and do their thing and create a gangrene. So that is just a special type of coagulative necrosis with superimposed saprophytic bacterial infection. Okay, then we come to the next thing. What time was this class to end? So that I don't overshoot the timing. Okay. Who is it there? Okay. Then we have a new concept. This new concept is called apoptosis. And apoptosis is a Greek word that means programmed cell off or falling off. The cell falls off. But in reality, it is a genetically programmed cell death. That after 120 days, the red cell must die off. After 6 hours, the neutrophil. After 14 days, the basal cell of the skin must be shed off as a superficial cell. So it is programmed that cells are not immortalized. And they are not immortalized because you have a physiological process where some are shed off and some must come in and replace Every time you chew food, every time you secrete secretions, some of the cells are programmed to die off. The blood, some old cells are removed, new ones come in, so that they are able to function and maintain the individual as they grow and as they age. This is called apoptosis, programmed cell death, and it occurs in embryogenesis. If you go back to embryogenesis, you recall that there was the story of two cells, an ovum and a sperm. It becomes a fertilized egg, and that then divides, becomes two cells, because mitosis forms two cells. Then the two become four, and it progresses like that until you see an embryo. And as this is happening, each of the cells that are being formed, some will be programmed to become the brain, the kidney, wherever. But not all of them will be eventually programmed into anything. Some of them must die off so that the rest of the cells continue and lead to whatever is programmed to be. So apoptosis occurs during embryogenesis, and it can also occur because of pathological processes. It is also seen in the cells that respond to hormonal stimulations. The endometrium must be shed off, cyclic shedding off. The endocrine organs must produce cells. They are stimulated to produce the hormones, and as they are produced, they proliferate and produce the hormones. So there's hormonal stimulation. And then there's also the defense mechanism in immunological reaction. The cells are programmed to encounter antigenic material, produce antibodies, sensitize T cells, and this do their job, and then they have to die off, and we move on to the next thing. We got exposed to the wild type of corona, then we got Delta, and as the cells were getting adjusted to the whatevers we are being exposed to, the ones that were not needed have to die off. Otherwise, we would get accumulation of lymphocytes within the lymph nodes and the marrow. And when that happens, that becomes another disease process. When this programmed cell death does not happen, then we end up with tumors. When it happens in pathological processes, we end up with diseases. So it occurs in embryogenesis, it occurs in hormonally stimulated organs, and it is also a defense mechanism in immunological reactions. It can also, it also occurs when cells are damaged by other injurious agents or noxious agents or other disease processes, and it also occurs in old age. Yeah, there's a shrinking in old age. The brain shrinks, the kidney, everything becomes smaller in old age. And the part of these processes include apoptosis. It is very similar to cell injury because it is removing cells. So it has similar processes, but it is an energy-dependent process. It is a deliberate process. Injurious process is accidental disease provoked, but apoptosis is programmed that after this period, this must happen. Otherwise, if it doesn't happen, that will be a disease process. So because it is deliberate, it is energy dependent. And the energy that it uses is the ATP. The ATP is what is needed for apoptosis to occur. When apoptosis does not occur, then we talk of a tumorous situation in some of the tumors that we see, like lymphomas. So we say it is seen in physiological processes. It is seen in adaptive processes. The cell changes, some cells have to be removed. And then it's also seen in pathologic processes. The cells are injured, but they have to come out. And therefore, it is seen across the board in embryogenesis and then also later in life. What we see on electron microscopy to differentiate it from cell death, or which is a cell necrosis. Actually, necrosis is actually just cell death during life. On electron microscopy, there's shrinkage of the cell. If it was 5 microns, it becomes 1 micron. So it shrinks and becomes smaller. And then, because of shrinking, the cytoplasm appear dense, so it stains more intensely, and the organelles end up being packed tightly because of that shrinkage. And also, it doesn't happen just in the cytoplasmic contents. It also affects the nucleic material where there's chromatin condensation. They come together and condense and eventually break up into fragments. And they don't, they also become almost cardiorectic, but they initially are big fragments. You can find 2, 3, then eventually becomes small and completely disappears without leaving a trace. So it does very much almost like what the cell injury is, but here there's the involvement of shrinkage. There's the contents diffuse out and the thing shrinks down. And there's formation of blebs also, and eventually little bodies that have nucleic and cytoplasmic contents that we call apoptotic bodies. So we see apoptotic bodies in pathological processes, physiological processes as well. And that is breaking off of nucleic material and cytoplasmic material with a cytoplasmic membrane and forming little bodies, almost like a small cell, but it's actually a apoptotic body. It's not a full cell, it's just components of it. They provoke phagocytosis, so they are taken up by macrophages, and we are able to see them ingested by surrounding cells or the macrophage, which come up to mop up the dying cells. The difference is that whereas in the necrosis, there's plasma membrane damage with the discharge of whatever was in the cytoplasm and the nucleus, but this one retains the nuclear membrane. We say that it's energy dependent. So not nuclear membrane, plasma membrane. It retains plasma membrane around this shrunken nucleic and cytoplasmic contents that may be a single entity or smaller fragments that are called apoptotic bodies. It can involve a single cell or groups of cells. Depends how fast they are proliferating or the disease process that is there. So you can see single cells or multiple or the apoptotic bodies that have been derived from the thing. And the most interesting part is that because it is an energy dependent intentional process, it does not provoke inflammation. Necrosis, on the other hand, provokes inflammation, and around a necrotic part, the surrounding living tissue provokes the next pathologic process you'll be discussing, which is called inflammation. But in apoptosis, we don't see that. Okay, so this is a summary of the differences. Just derived from Robbins. You will be able to see that when you read for yourself. The differences in the various things that we see between the two. So when it comes to cell size, there's enlargement of the cell in necrosis because we get fluid. There's a loss of, it can't maintain the gradient. There's an energy being produced. So fluid comes in and there's swelling. But in this other one, it's the opposite. There's shrinkage. Yeah. There's no loss of the plasma membrane. Instead, it's diffusion of the contents, and therefore you get shrinkage. The nucleus becomes pycnotic, or we see pycnosis, or karyorectic, or even complete dissolution. Whereas in this other one, you don't see as the nucleus on its own. It carries some cytoplasmic content. So we see nucleosome-sized fragments or karyorectic fragments. They still have cytoplasmic contents around them. Whereas this one, the cytoplasmic content has gone. You're just seeing the nucleic material dissolving, breaking up, or completely, or shrinking. Pycnosis is shrinking. The pycnosis is different from the apoptotic bodies because pycnosis doesn't have cytoplasmic material. Okay, plasma membrane, we've already mentioned this one, it is disrupted. This is intact, or it might be altered, or different orientation of the lipids. How about the cellular contents? There's enzymatic digestion because the lysosomal plasma membrane is damaged, release of the enzymes, and damage of the cell. Whereas this one, they're intact. The plasma membrane is the trick. It remains intact and they are released as apoptotic bodies. Provokes inflammation, does not provoke inflammation. Physiological or pathological in its causation, whereas this one is quite variable. Embryogenesis, pathologic, but also related to other, it's pathologic, but whereas this other one, you have all sorts of issues coming in. Physiological, embryogenesis, hormone related, generally to be able to maintain some of this. Of course, it can also be pathological in tumors. We also see it in tumors, but often physiological and rarely pathological. Okay, and with that, I come to the end of this cell injury. So we'll come back. Okay. I see there's quite a bit in the chat. And I'll try to answer some of these. Examples of apoptosis. When we have, when the lymphocytes are proliferating to form memory cells, the rest must be finished so that you remain, they do their job and then the rest must be finished and only remain with memory cells. If you continue retaining all those cells which came into play, then it would end up being a lymphoma. Even in lymphomas, we see proliferation and see apoptotic bodies where the cells are also being whatever. So apoptosis mainly in embryogenesis and when making memory cells in immunology, yeah. You counter an antigen, the cells proliferate, make memory cells, they go and reside in the lymphoid tissues, and the rest die off, yeah, through that process. What was the other? Do all diseases lead to cell death? No. They will only lead to cell death. Even you can get Staph aureus, but you don't get a boil because it doesn't become too bad. You just get a few cells dying off and everything is restored. But if the bacteria proliferate and cause a lot of damage, then you will end up with quite a bit of cells dying off. Not all of them will cause cell death because some cause irreversible, they cause reversible injury. When they cause irreversible, you see, we're able to see necrotic areas because you have big spaces, you have many cells affected. Individual cells might be affected and they die off and they're replaced. You will see when you're learning about healing and repair, they'll be replaced either by regeneration or by fibrous tissue. So as the bacteria or whatever injurious agent one is encountering, not all cells will undergo necrosis. We are able to see necrosis when many cells undergo that process. Otherwise, these are individual cells, but they may not affect the actual functionality, but they may affect the biochemistry in such a way that we are able to see. A person drinking alcohol injures liver cells, some die off. We see a bit of that effect in the biochemistry, but we don't see the sum total effect in the organ. We might see an individual cell which died off, but we might see one that is recovering. So it is how severe the injury is to the cell, how severe the injury is to the cell. I think I am not sure that I've answered what you are looking for. Inflammation is a separate topic. If you look at the topics we gave you, cell injury, inflammation, healing and repair, cellular adaptation, all those, and you'll see how they tie in. Cell injury provokes the surrounding tissue to mount a physiologic process called inflammation. Inflammation tries to contain the spread of that injurious agent, but in the process creates a disease process. Yeah? Cell injury will make patients come to hospital. The physiological process, inflammation, will also make them come to hospital. So all these topics we shall be talking about, we are showing you what is happening in the body, and each one of them is capable of causing a disease process. Yeah? It's capable of causing a disease process. The healing process can be severe so that people end up with keloids and they have now to come and have them cut. So they're split into topics, but they merge into each other. It's a continuation. The body doesn't stop at cell injury. It goes to the next process, which is inflammation. And when it deals with the inflammation, there's the healing which kicks in, and that's how things happen. If the healing fails, it can even induce a neoplastic process, a tumor. So all those will come as individual entities, despite the fact that we are introducing you to new terminologies. You will learn a lot of vocabulary that you'll be learning. Resolution you will hear in healing and repair when everything is restored back. Of course, the dead cells were digested and removed, but a new cell similar to it comes and replaces it. That is the resolution. But the dead cell was removed. If it is too much, then even when it's removed, it won't get back the normal cell. Instead, it's replaced with fibrous tissue, and you see a scar. So when you see somebody with a scar, you know there had been a cell injury there, maybe a cut, maybe an abscess, whatever it was, and healed with a scar. Some had it but didn't leave a scar because there was a resolution. Yeah, I hope that has helped whoever asked that question. So it's a continuum. Yeah, cardiolipase is the last. No, it starts, you start digesting the nucleic material until it disappears completely. You start the feeding basophilia. Basophilia is because the nucleic material is being broken down. And when it's broken down completely, then it disappears completely. But it can be fragmented and then dissolved completely. Okay. Okay, so that is all right. Read the textbook, a lot of new things. Of course, if you can stop some of these things we've been talking about, then you are able to deal with the disease processes and the patients are happy. You are able to stop the next consequence and the patients are happy. If we were able to stop the corona at just the upper respiratory tract level, then we wouldn't have the damaging effects in the chest, in the lower respiratory tract, where it was causing necrosis. It was damaging the alveoli. As some of the survivors have fibrosis. Yeah? If you have fibrous tissue in the lung, it won't function. If we had caught it at the level, the site of entry, then we would have been able to deal with it and do that. What has happened, the host immunity, the viral transformation have modified the disease, but the initial one was quite severe. Okay, so we shall meet next. When we have the next topic, we have a lot to discuss over the coming academic, over the next weeks of the current academic year. And if you have issues, please let us know. We are just readjusting to the timings and accommodating everything that we need to accommodate. Some topics are very long, so they'll be able to utilize all the time without being rushed. If we find that it is possible to have breaks in between, then we can arrange so that we have a small break between the first part of the lecture and the next part of the lecture. We used to have 2 hours, now we've been added, I think we have 3 hours. So we will be adjusting to accommodate a short break. Okay, thank you. That's it.
Thank you.